By producing proteins that may be able to treat diseases like cancer and COVID-19 in a fraction of the time it takes with conventional approaches, Google DeepMind's new AI is revolutionizing the area of protein design. AlphaProteo, an AI model, creates proteins that bind to particular targets, expediting the process of researching diseases and finding new drugs. AlphaProteo is revolutionizing biotechnology and medicine with its unprecedented capacity to outperform proteins created by humans. Kindly spend a few seconds subscribing for more updates before we begin. A novel artificial intelligence system creates proteins that bind to target molecules successfully, potentially improving medication development, our knowledge of diseases, and other areas. Protein interactions are essential for all biological processes in the body, including immunological responses and cell proliferation. One protein can bind to another like a key does to a lock, aiding in the regulation of important biological functions. Although we have already gained a great deal of understanding from protein structure prediction tools like AlphaFold about how proteins interact with one another to carry out their tasks, these techniques are unable to directly modify these connections by creating new proteins. On the other hand, scientists can produce new proteins that bind to target molecules with success. These binders can expedite research in several areas, including drug development, illness understanding and diagnostics, imaging of cells and tissues, and even crop resistance to pests. Although there has been significant progress in recent years with machine learning approaches to protein design, the process remains tedious and necessitates a large amount of experimental testing. They are pleased to present AlphaProteo, their first artificial intelligence (AI) system for creating unique, powerful protein binders that may be used as building blocks in biological and medical research. This technique could expedite our comprehension of biological processes and facilitate the creation of biosensors, new drug discoveries, and other endeavors. AlphaProteo is able to create unique protein binders for a range of target proteins, including VEGF-A, which has been connected to diabetes issues and cancer. An AI technology has effectively designed VEGF-A protein binders for the first time. On 7 target proteins that we investigated, AlphaProteo also outperforms the best current techniques in terms of binding affinities and experimental success rates, ranging from 3 to 300 times better. With more than 200 million items in the most recent database release, UniProt, the common repository for protein sequences and annotations, is widely covered. The human proteome and the proteomes of 47 other major organisms that are crucial to study and world health are available for individual download from them. Additionally, they offer a download for the UniProt, Switzerland Prot, hand-curated subset. AlphaFold produced highly accurate predictions and was by far the most ranked protein structure prediction approach in CASP-14. Even though AlphaFold currently has several drawbacks, the CASP results indicate that it has the potential to improve biological research and help us better understand protein structure. Being eager to know the intricate ways in which proteins bind. It is challenging to build protein binders that can firmly attach to a target protein. Conventional techniques take a lot of time and include several rounds of labor-intensive lab labor. Following their creation, the binders go through many experimental iterations to maximize their binding affinity and ensure that they attach enough tightly to be of any utility. With over 100 million predicted structures from AlphaFold and a ton of protein data from the Protein Data Bank, PDB, AlphaProteo has been trained to understand the various ways that molecules might attach. AlphaProteo creates a candidate protein that attaches to the target at certain spots based on the structure of the target molecule and a set of preferred binding positions on the target molecule, proving effectiveness on crucial protein binding targets. According to Google DeepMind, we have created binders for a variety of target proteins to test AlphaProteo, such as the viral proteins BHRF1 and SARS-CoV-2 spike protein receptor binding domain, SC2RBD, and the 5 proteins IL, 7R, PDL1, TRKA, IL-17A, and VEGF-A, which are involved in cancer, inflammation, and autoimmune diseases. Best-in-class binding strengths and extremely competitive binding success rates characterize our method. When it is tested experimentally, AlphaProteo produced in silico candidate proteins for 7 targets that bonded tightly to the target proteins. What we should understand about AlphaProteo. The 2 components of AlphaProteo are a generative model trained on structure and sequence data from the Protein Data Bank, PDB, and a distillation set of AlphaFold predictions. It also has a filter that assigns a rating to designed designs based on how likely it is that they would work in experiments. After researchers input the structure of the target protein and, if desired, identify hotspot residues that correlate to the target epitope, the generative model generates the structure and sequence of a potential binder for a target. Researchers generate a large number of design choices and then narrow them down to a more manageable set before starting any experimental testing. The generative model outperforms the state-of-the-art method based on in silico benchmarks, real-world protein binding with AlphaProteo. The Francis Crick Institute's research teams assessed and validated AlphaProteo, a protein binder, in silico in a wet lab. It was found that the binders, which included SC2, RBD, and VEGF-A, had advantageous biological roles and binding interactions that agreed with AlphaProteo's predictions. AlphaProteo may be able to reduce the number of preliminary protein binder application tests based on its findings. However, it was unable to produce TNFA binding proteins that are beneficial against autoimmune diseases like rheumatoid arthritis. Since producing proteins for useful applications only entails achieving strong binding, the team aims to improve AlphaProteo's capacity to tackle challenging targets. Progressing protein science cautiously. Protein design is a rapidly evolving field of technology that has enormous potential to advance science in many areas, including the detection of disease drivers, the development of diagnostic tests for viral epidemics, the promotion of environmentally friendly manufacturing methods, and the elimination of environmental pollutants. To support local efforts to develop best practices and to help design their phased release approach for this work, Isomorphic Labs is working with outside specialists. Google DeepMind is looking into the influencing biology challenges and limitations of Isomorphic Labs' drug design software, AlphaProteo. To offer a responsible protein design service, the firm is also expanding the range of its design capabilities, working with researchers in the fields of machine learning, biochemistry, and structural biology, and improving the success rate and affinity of AlphaProteo's algorithms. Following a single round of medium throughput testing, AlphaProteo, a unique AI system for drug development, has proven to be able to generate sub-nanomolar binders for a variety of targets. These highly expressed, thermostable, and minuscule binders, with a molecular weight of 5 to 15 kDa, are already suitable for some research applications. A target crystal structure is used as input for all binders. Nevertheless, the research has only validated a limited number of targets. The company wants to improve AlphaProteo's capabilities and address a wider range of binder design issues, which may result in the development of cutting-edge solutions for biological applications, including imaging of proteins, cells, and tissues, and regulation of cell signaling. Let's validate the results. Numerous studies have been conducted, and the results have shown that these binders' binding interactions did indeed resemble those anticipated by AlphaProteo. Furthermore, the teams verified that the binders serve beneficial biological purposes. For instance, it's been demonstrated that a few of our SC2 RBD binders stop SARS-CoV-2 and some of its variations from infecting cells. Based on its performance, AlphaProteo has the potential to significantly cut down on the amount of time required for preliminary studies utilizing protein binders for a variety of applications. We are aware of the limitations of our AI system, though, since it was unable to create effective binders against TNF, a protein linked to autoimmune illnesses such as rheumatoid arthritis, which is the 8th target. We chose TNFA as a robust challenge for AlphaProteo because computational study indicated that designing binders against it would be quite challenging. We'll keep enhancing and growing AlphaProteo's capabilities so that it can eventually handle such difficult objectives. In the process of researching and developing proteins for potential practical uses, strong binding is typically just the beginning of the process. There are still several bioengineering challenges to be solved. Okay, let's move toward the direction of responsible protein design development. Protein design is a rapidly developing field of study with enormous potential to advance scientific knowledge of everything from the causes of disease to the development of diagnostic tests for viral outbreaks, more environmentally friendly manufacturing methods, and even environmental pollution removal. Building on our long-standing commitment to accountability and safety, we are collaborating with top outside experts to help shape our phased dissemination strategy for this work and contribute to community initiatives aimed at creating best practices, such as the Nuclear Threat Initiative's (NTI) recently established AI Bioforum to take into account potential biosecurity risks. In the future, we will collaborate with the scientific community to apply AlphaProteo to significant biological issues while being aware of its limitations. At Isomorphic Labs, we've also been investigating its potential for medication design, and we're enthusiastic about what lies ahead. In the meantime, we're working with researchers in machine learning, structural biology, biochemistry, and other fields to develop a responsible and more extensive protein design offering for the community. We're also expanding the range of design problems that AlphaProteo can handle and improving the success rate and affinity of its algorithms. Thank you for watching. Please use the like and subscribe buttons for additional videos if you liked this one. Don't forget to switch on the notification bell to receive notifications when new videos are uploaded.